Welcome to homework.
Hello student, here the question is given and the question is about the expression of the gene. So here the expression of the lo1 gene, and this is in case of corn pollen. So here we have to regulate this gene expression until it's needed during germination. So we have to regulate this one. So we have to propose the mechanism here. So we can say the gene regulation, if we are talking about this, can be occur at a different level and mostly included at this transcriptional level, and another one is the post-transcriptional. So during the transcription simply means the formation of from the DNA to mRNA, and in this case post-transcription is the after the synthesis of these mRNA. So here after mRNA synthesis. So at these two levels the gene can be regulated. So if we are talking about these first one, which is the transcriptional repression. So in this case the one could be method, and this is the protein promoter methylation. So we know for the transcription of any gene, the promoter region is the one which is available for the RNA polymerase. So these promoter region of the lop1 gene could be methylated in the pollen cell before they are released. In pollen cell before they are released. So in that case DNA methylation is taking place, and this is the most common method of epigenetic modification by DNA methylation or promoter methylation. So that can repress the gene expression by preventing the binding of transcription factor to the promoter region. So if transcriptor factor doesn't bind to the promoter, then the transcription is not going to take place, so the repression will be happen. And after this transcription factor inhibition, if these factor inhibited, then it is only activated upon contact with the corn silk. Until the interaction occur, the transcription factor may be inhibited, so preventing them from the binding to the gene promoter, and the transcription is not going to initiate until now. And the second one, which is the one is the post-transcriptional. So in case of post-transcriptional repression, we know the most common one is the microRNA or miRNA. So these miRNA are the small RNA molecules, and these can bind to the mRNA. So it can bind to mRNA, and after binding, degradation and inhibition of the translation, degradation or inhibiting the translation process. So this mRNA or the microRNA specifically expressed in a pollen, and actively it is going to repress the translation of these mRNA of loop gene. So this is after synthesis of mRNA. And next one, it could be the another one is RNA binding protein, binding proteins. So these also could bind to the lop1 mRNA and prevent translation or stability of this product of this particular gene. So in both the cases, the expression of these lop1 gene is going to be inhibited. So if we want to draw this, so suppose this is the promoter region, and after promoter there is the gene. So this one is the lop1 gene. So these promoters region, if it is methylated, so due to methylation, the transcription factor is not able to, suppose these are the transcription factor, it is not able to bind with this. So the expression or the synthesis of these lop1 is going to be inhibited. So here this repression of this lop1 is done in one case, and another one, if these lop1 is synthesized as mRNA, then these mRNA need to be bind with the another microRNA, which is the small RNA. So after binding, these are degraded. So the further protein synthesis or translation is inhibited. And this is the case of post-translational modification. And another one, after protein synthesis, ribosome binding protein is also going to inhibit this translation of the particular protein. Hope you found this video useful. Thank you.